Thank you very much, Ahanjit, for agreeing to give a Build a Cell seminar, and you can get started.
Hello, everyone. So first of all, I would like to thank Kit for inviting me to give a talk at this very prestigious community of artificial cell researchers. So the very first time I heard about Build a Cell, I was very excited about this idea because when I was in my doing my PhD at UCSD, I was a part of a small group which was a MURI collaboration between 6 groups across different universities in the US. And I thought that that was a very cool idea because that brought people from many different expertise. Like some of us were chemists, some biochemists, some chemical engineers, some physicists, and we exchanged different kinds of ideas when we met. But this is a much bigger group of much more diverse people. So I'm pretty sure that everyone who attends these meetings, they take away very interesting ideas from here. So I will start with a little bit about myself. My name is Ahanjit Bhattacharya. I am an assistant professor of chemistry at the Indian Institute of Science, Education and Research in Bhopal. So I very recently started my own lab from September. Before that, I was in US for 10 years. So I'm a chemist by training. I had my undergraduate education at the Indian Institute of Technology, Kharagpur. And then I moved all the way to San Diego, where I did my PhD at the University of California, San Diego. And then I was very much in love with the nice weather and views of California. So for my postdoc, I just decided to go a little bit up north to Stanford, where I spent 4 years as a postdoctoral scholar. And then after my long stay in the United States, I finally made it back to India. And right now I'm in Bhopal, which is also known as the City of Lakes. It's a very beautiful city located at the heart of India. And the name of my institute is Indian Institute of Science, Education and Research. So this is a part of the ISER system. So there are 7 of these all across India, and they are focused on basic sciences. And these are not much old as compared to the IITs, but they have already established themselves as leaders in basic sciences in India. And I believe that they're pretty well known all across the world. So this is my lab's webpage. So I decided to name my lab as the Chemical Synthetic Biology Lab. This is a QR code where you can take a further look at my lab's website. So I will start with this phrase, which I also put on the title of my talk, like chemical synthetic biology. So I will tell you like what exactly I meant by that and where I first heard about this phrase. So the reason why I started using this phrase is because I think this phrase captures the style of my work in the most perfect way. And I first read about this phrase in a mini review by 2 of the stalwarts of the artificial cell field, Pierluigi Luisi and Pascal Stano. So they use this phrase chemical synthetic biology in the title of this review article. Now, I didn't find this phrase being used very much, although synthetic biology is a very well-known term. But this phrase, chemical synthetic biology, I didn't find the usage of it too much in the literature or in ordinary usage. But I felt that this is the term which perfectly captures the kind of thing I do. So I will tell you what exactly I mean by chemical synthetic biology. So the premise of chemical synthetic biology sits on a few fundamental questions like what is life and how did it originate, and then how we can think of a minimal cell and a minimalistic representation of a biological process. Now, like any line of thought, there is a historic progression to it. So I will give you a little bit of that historic perspective to what is this style of chemical synthetic biology. So first of all, let's go back in time to 1953, when a 21-year-old PhD student named Stanley Miller did an experiment which stunned the scientific community of his time. So Stanley Miller was a PhD student of Harold Urey, a Nobel laureate, and he was trying to understand how the building blocks of life were formed in the first place. So he made his own glass apparatus and filled it with some gases which were likely present in the atmosphere of a primitive Earth. And then he passed electricity through that for 7 days. And in the end, he got a mixture of simple amino acids and organic molecules. Now, this was the first experimental demonstration of the idea of abiogenesis, or in other words, he showed that the building blocks of life can be formed from very simple molecules that can be found in atmosphere or in the environment. Next, I give this example from 1970, where Hargobind Khorana's group tried to understand how genes function. So what they did is that they chemically synthesized a gene. Now, this is a gene for a tRNA, and showed that the gene can transcribe its product if it's added to a cell-free expression system. Then in 1991, Eckhard Wimmer's group wanted to understand whether viruses need a cellular host to replicate. So before that, the idea was that a virus certainly needs a cellular host for its replication. So they challenged the idea, and what they did, they took the DNA for proteins which code for poliovirus and added to a cell-free expression system. And they showed that the protein got expressed and whole virus particles sprung out of that mixture. So everything took place completely outside of the cell. Now in 2001, Takwe Ueda's group took a tour de force exercise to understand what is the minimal requirement for protein synthesis. Yes. So they came up with something known as the pure system. So the pure system is very familiar to the synthetic biology community. So it's a well-defined mixture of all the necessary components like proteins, tRNAs, small molecules, ribosomes, and so on, that is essential to synthesize a protein outside of cells. Now, this is, as I mentioned, a very useful tool in synthetic biology, and I also have used it quite often during my own research. Now, from 2000s onwards, there has been a significant drive to develop something known as an artificial cell. Now, this community certainly doesn't need a fresh introduction to artificial cell, but just making a quick definition of it. So it's a lifelike system or entity that's built from fundamental chemical and biochemical building blocks, and one that shows some basic features of cells, like presence of compartments, presence of some metabolic reactions, growth, division, and so on. So this is where my stories start. So my first story is about building artificial cells and organelles from fundamental building blocks. So throughout my research, I have worked with different kinds of lipid molecules. Now, some of these lipids have one polar head group and one hydrocarbon chain, like oleoyl thiogalactopyranosides. Then many of the lipids that I've worked with have one polar head group and two hydrocarbon chains. These are probably the most common types of lipids found in nature. So in this example, I show a lipid known as an ester analog of sphingomyelin, which I found through a very interesting esterification reaction catalyzed by metal ions. And during my postdoc, I worked on some very interesting looking lipids. So these are bipolar lipids. They have two polar head groups and a continuous hydrocarbon chain in the middle. So they also have multiple branches, and these are typically found in extremophilic archaea. Now, in spite of differences in their chemical structure, so all these lipids self-assemble to form these spherical compartments, which are about 1 to 20 microns in size, known as giant vesicles. Now, these giant vesicles are commonly used to mimic the overall membrane-bound architecture of cells. Now, I was working with many of these lipids, and one of these lipids was galactose oleoyl amide, or GOA. So I was trying to form some vesicles with these single chain lipids. Now, quite accidentally, I found that if I mix this lipid with non-ionic detergents like IgPal, then it forms a turbid dispersion with some dense spherical droplets in it. Now, in the beginning, I thought that maybe these are multilamellar vesicles because sometimes they also look like these dense sphere-like objects under the microscope. But I noticed some unusual features of this. So when these droplets met, they often fused to form a larger droplet. And this turbid dispersion that you can see in the picture, it settles to form a single dense phase at the bottom of the tube. Now I found that these droplets are formed over quite a wide ratio of proportion between GOA and IGPAL, like typically 5 to 1 to 1 to 1. So this kind of liquid-like property reminded me of something. So the first question that came to my mind is that are these droplets some kind of liquid-liquid phase-separated system or quasar? Now I was very much interested in origins of life. So I was familiar with Alexander Oparin's idea that the earliest forms of cells were probably quasar. So these are these spherical aggregates formed from protein-like material. And back in 1920s, Oparin proposed that these were the progenitors of all forms of cells. Now the quasar theory sort of remained in scientific backwater for most of the history until the early 2000s when these kind of structures started to be discovered in cells. So these are now known as biomolecular condensate, and I believe this is still a very active field of research. Now, these kind of structures, which I just referred to as quasar, were not very frequently encountered with lipids and surfactants. There were just some reports here and there, like these droplets which were formed from this surfactant known as AOT. But other than that, there were not a lot of reports. So anyway, we started doing a lot of physical characterization experiments with the droplets that we observed and found that the GOA and IgPL droplets indeed display hallmark characteristics of lipid quasar, which are also referred to as lipidic sponge phase. So I will walk you through some of these characterization techniques that we did. So first of all, we took the droplets and did small angle X-ray scattering. So in both the dispersed phase and the coalesced phase, the droplets gave a broad intensity profile like this. Now this suggests that these droplets do not have any long range order, but they are disordered liquid-like material. Then we took the coalesced phase from the bottom of the tube and did something known as thermogravimetric analysis. So here we heat the sample from room temperature to a much higher temperature. And we noticed that by the time the temperature was about 100 degrees Celsius, there was about 70% loss in mass. So this lost mass likely comes from the water that's present in the sample. So these are not simply oil in water kind of droplets, but aqueous two-phase droplets. Then we took the sponge phase and then we did freeze fracture cryo scanning electron microscopy. And then we could see that they have a porous interior. Then we did cryo-TEM of the droplets, and we could see that they have a network of bilayers in it. So combining all the characterization data, we came to the conclusion that these droplets indeed belong to something known as a lipidic sponge phase. So what is this lipidic sponge phase? So you may think of this as a three-dimensional network of surfactant bilayers that are crisscrossed by aqueous channels about 10 nanometers in size. Now, I was made by two science artists, and this was published in a science art journal known as Leonardo. So this animation shows how a bicontinuous structure, so that's one of the technical terms used for this kind of a structure, may look like on the inside. So all these interconnected bilayers that we are seeing here are likely very dynamic, and their connections are forming and reforming all the time, and the pores are changing in sizes constantly. Now, we were also very excited to notice that this kind of network of membranes can be found inside of cells, particularly in the smooth endoplasmic reticulum of plants. So looking at this kind of apparent similarity, we decided to call the sponge phase droplets as mimics of membrane-rich organelles like the endoplasmic reticulum. So I was very excited to figure out like what these structures were and what kind of nanostructures they may have on the inside. I started exploring the physical chemical properties of the drop. Now, first of all, I found that these droplets are formed very robustly in many kinds of common buffers, pH, temperature, and even in very complex heterogeneous media like seawater or in cell culture media like DMEM, FBS. Now, the droplets, they have affinity for molecules which are hydrophobic or somewhat hydrophobic. So in this very nice looking palette formed from different dyes, what I want to show that the dyes which are somewhat hydrophobic, they're very nicely retained inside the droplets. Now, because these droplets are sponge-like, they don't have a strict boundary like vesicles do. So if you add some dye from the outside, they will go through the pores, and if they have affinity for this material, they will sit there and get retained there. Now, this is not the case for every dye. So dyes like Arexophore 488, which have multiple charges on it, they are not retained inside the droplets. And you can see that the droplets look somewhat dark against the background. Now, this is also useful because we may use dyes like Arexophore 488 to selectively label some macromolecules if we want to study their partitioning behavior inside the droplets in an unambiguous manner. Now, because the droplets are composed of a lipid which has a galactose head group, we naturally tested whether they have affinity for proteins which have affinity for galactose. And I found that, yes, many proteins which have affinity for galactose do get partitioned to the droplet. So some of these proteins are leg A, it's a lectin from Pseudomonas. Galactin 3, it's a human or mammalian protein. And LAC-I, it's a bacterial protein. So the fluorescent analogs of this protein, they're all very nicely retained inside the droplet. Now, sometimes we could see some interesting things under the microscope. For example, if we added excess of this galactophilic protein like A to the droplets, then they started making some transformations in terms of shape. So here you can see that these droplets seemingly start splitting into two daughter droplets, and this kind of an event um, gives an impression of some kind of cell division. Okay, so next we wanted to know whether these kind of interactions are retained in a more complex media. So what we did here is that we took pure system. I mentioned the name of pure system a little bit ago. So here we took the DNA that codes for this protein SFGFP LAC-I and added the droplets to the pure system and let this protein get expressed in pure system. So in this video, you will see that SFGFP LAC-I gets expressed in pure system and it spontaneously gets uptaken inside the droplets as it gets expressed. Now, it's also important to notice that this experiment takes place over quite a long time, and the droplets do maintain their structural integrity over this period of time. And just like organelles uptake specific molecules inside them, these droplets also soak up this very specific protein out of this complex mixture as it gets synthesized. So this is another demonstration of some kind of an organelle-like function of the sponge phase droplet. Now, it's not just hydrophobic molecules and galactophilic proteins. The partitioning properties of the droplets can be programmed if we add certain amplifiers to them. So when we add his-tagged SFGFP, then the protein doesn't have any specific affinity for the droplets. So you can see that the fluorescence is everywhere. But when we dose the droplets with a lipid which has a nickel MTA group on it, then the nickel MTA group binds to the histidine groups of GFP, polyhistidine group of GFP, and then the protein is nicely partitioned inside of the droplet. And using this kind of a doping strategy, we found that we can achieve very high concentration of protein inside the droplets, reaching almost 1 millimolar, which is roughly the order of concentration of proteins that is found inside of cytoplasm. And what's also interesting is that this kind of a doping strategy can be made quite generalized. So we found using different kinds of affinity groups that we can direct the partitioning of proteins or other macromolecules in the droplets in a programmable way. So next, I will give one example where we showed that the droplets do have some selectivity in terms of the size of the molecule it can partition as well. So what we did here, this work was done by one of my lab members, Christy Cho in Debra's lab. So what Christy did is that he took fluorescently labeled DNA of different sizes and had those DNA bind to the droplet through streptavidin and biotin interaction. So up to about 108 base pair of DNA, the DNA gets very nicely partitioned inside of the droplets. But as the size of the DNA increases, then they are no longer as nicely retained inside. And the reason is that the droplets do have a certain range of size of the aqueous channel. So when the DNA gets too big, like about at 308 base pairs, you can see that they're no longer inside the droplets, but they form some kind of corona around the droplets. So they are excluded from the pores that are present inside the droplets. And above these things, the DNA is not retained inside the droplets at all. So next, I will describe one experiment where the property of programmability of droplets with respect to affinity groups and also size were demonstrated at the same time. Now, this beautiful experiment was done by my colleague, Henrike Niederholzmeier, who is the professor at U Munich right now. So in this experiment, we took a protein known as 5B. It's a plant protein, and 5B stands for phytochrome B. Now, 5B can bind to a partner known as PIF. Now, in the binding conformation, 5B binds to PIF, but if 740 nanometer light is shined, then it changes to a non-binding conformation, and PIF is released from the binding site. Now, if the wavelength of light is switched back to 660 nanometer, then 5B changes to binding conformation, and it can bind to PIF again. Now here, PIF was fused to SFGFP and SSRA. So I will tell you in a bit what this SSRA is about. But before that, I will show you how this switching of wavelength leads to very predictable uptake and release of PIF, SFGFP, SSRA from the droplets. So in the beginning, when the 740 nanometer light is on, you can see that the droplets are dark. And then when the wavelength is switched to 660 nanometer, 5B changes to binding conformation, and PIF, SFGFP, SSRA goes inside the droplet. And then again, when the wavelength is switched, the protein goes out and it goes back again when the wavelength is changed again. Now, we took a protease known as ClipXP. Now, this is quite a large protein complex, about 600 kilodaltons in size, and its dimension is about 14 nanometers. So this is larger than the typical pore size of the droplet, which is about 10 nanometers. So ClipXP will remain outside of the droplet. Now, ClipXP can chew up any protein which has this SSRA tag on it. So the idea here was that when 5B is binding to PIF, SSGFP, SSRA, then it's inside the droplet and it's protected from ClipXP. Now, when the wavelength of light is changed to 740 nanometers, then PIF, SSGFP, SSRA is outside of the droplet, and then it can get chewed up by ClipXP. And then again, when the wavelength is switched back to 660 nanometer, then it goes inside the droplet again, and it gets protected from ClipXP. So we were actually able to do this switching of wavelengths and control the degradation of protein inside and outside the droplets over a few cycles. So when the protein is outside, you can see that the fluorescence goes down quite rapidly, and then the slope is decreased, and then it goes up again, and then again it's somewhat flat and goes sharply again. So this is again an organelle-like function of the droplets, where specific molecules are written in the inside, and when the molecules are released, then a particular biochemical like reaction, in this case, degradation of a protein takes place. Now, because the droplets are composed of amplifiers, we naturally ask if they have affinity towards membrane proteins because they are also amplifiers. So we found that membrane proteins like cytochrome C oxidase can be nicely partitioned inside the droplet, and also the protein is functionally active. We found that it can readily oxidize its substrate ferrocytochrome C to ferricytochrome C. So next we did a more complex experiment. So in this case, we decided to express a membrane protein in pure system in presence of the droplet. So we took a membrane protein enzyme known as diacylglycerol kinase, this is from E. coli, and added the DNA that codes for this enzyme to pure system in presence of the droplet. So we found that indeed this enzyme is functionally active. It got expressed, as we can see that when we added the DNA for a GFP fused version of this protein, the fluorescence can be detected inside of the droplet. And from LCMS measurements, we saw that the protein catalyzed the phosphorylation of its substrate, this is monoolane, to the corresponding phosphorylated version. So here, this is the peak coming from the starting material, and it's gone after 3 hours, and the product shows up, and the starting material is consumed entirely. So with this, I would like to wrap up my first story. So here I talked about how different lipidic structures like vesicles and then sponge-phase droplets can be used to mimic the basic membrane-bound architecture of cells and certain organelles. Now, the sponge phase is a very little class, explored class of self-assembled materials, and there are many fundamental questions that needs to be explored, and these are some of the things I will be interested in exploring as an independent researcher. So for example, what are the general rules for designing sponge phase lipids, and then how we can predictably control the pore sizes of sponge phase, and then how we can improve the stability of sponge phase against coalescence, or make a monodispersed sponge phase droplets. And there is one idea that is I'm quite interested about is coming back to this concept of protocells in the context of origins of life. So as I mentioned that like in the origins of life, coacervates were considered as an interesting model of protocells. So my thought is that maybe these sponge-like structures can be a very interesting models of protocells because they are open, they're porous, they can exchange materials with the surroundings freely. So they could be a very interesting model where like the origins of life took place. So now we'll move to my second story, and this is about synthesizing new compartment material for artificial cells. Now, this story starts with a fundamental question, and this question is, do all membranes come from pre-existing membranes? And let's see where this question ultimately came from. In biology textbooks, we read about this famous statement, omnis cellula is cellula, by Rudolf Virchow, which means all cells come from pre-existing cells. Now, on similar lines, the membrane biologist Gunter Blobel wrote, omnis membrana and membrana, meaning all membranes come from pre-existing membranes. So let's see why this statement was made. So if we look at the way membrane lipids are synthesized in cells, then we will understand like why this kind of a statement was made. So in cells, the phospholipids are synthesized by enzymes, many of which are transmembrane proteins themselves, which means these proteins themselves need pre-existing membranes to fold and function properly. So now the question arises, if the machinery that makes the cell membrane itself needs pre-existing membranes to fold and function properly, how were the first membranes formed? And then, particularly how the first membranes were formed by macromolecular catalysts. And also, if we think of an origins of life kind of scenario, where it is thought that the earliest catalysts were ribozymes, which are catalytic RNA molecules, then it's hard to conceptualize that those RNA molecules will behave in the same manner as transmembrane proteins. So can we conceptually demonstrate that a soluble macromolecule, like a protein enzyme or ribozyme, can drive membrane lipid synthesis? And if we can do, then we can have some idea of how the earliest membranes may have formed. So my PhD advisor, Professor Neil Devraj, when he started his lab, he started exploring this question. And the first attempt to this question was designing chemoselective ligation chemistries that replaced some of the chemistries or biochemistries that took place in cell membranes for synthesizing a two-chain lipid. So what Neil started doing in the beginning is that he took two non-membrane forming amphiphilic precursors, and due to some chemoselective ligation reaction between them, they formed a two-chain lipid which self-assembled to form a membrane. Now I will give a brief demonstration of this idea. So this is something I did in the beginning of my PhD. So here I took a lipid fragment which has a histidine group on it, and this reacted with another lipid fragment which had a thioester group on it. Now this reaction is known as histidine ligation. It's sometimes used in peptide ligation chemistries. It's not a great reaction, but here we found that in amphiphilic media, this reaction was quite efficient, and it formed this phospholipid analog with a histidine group on it, and this self-assembled to form membranes in situ. And there were several other demonstrations from News Group in like about 10 years or so ago. Now, then I started to think that how can I bring a macromolecular catalyst in this picture? So either a protein enzyme or a ribozyme. Then I came across a very interesting enzyme known as FADD10. So this enzyme is from Mycobacterium tuberculosis. And what it does is that it takes a fatty acid and it converts the fatty acid to something known as an adenylate derivative with the help of ATP and magnesium. Now, an adenylate can react very rapidly with a primary amine group. So I designed a lipid fragment which has a primary amine group on it, and these two reacted very rapidly and chemoselectively to form this two-chain lipid with an amide group in it. And this two-chain lipid, as it formed on course of the reaction, it self-assembled to form vesicles, and you can see that the vesicles became more numerous, and they also increased in size. Now, next, I wanted to link the transcription and translation of FADD10 in pure system so that we could synthesize these membrane-forming lipids in sort of one-pot fashion. So the idea was something like this. We'll take a DNA that codes for FADD10 and let it express in pure system and have the lipid precursors present in the pure system at the same time. And then this enzyme will drive the synthesis of lipid from the lipid precursors, and the lipid precursors will self-assemble to form vesicles. However, we encountered some difficulties in carrying out this reaction with FADD10. So we found that when we put everything together, then the yield of lipid synthesis was not good. And we identified a few likely reasons for that. Now, first thing is that the fatty acyl adenylate that's generated by FADD10 has a low steady state concentration. And so the concentration at any point was not enough to have a sufficient conversion to the desired product. And also these aminolysed lipids that we are using, they were hydrolytically unstable because the pure system has many amphiphiles and it just cleaved at this linkage. So that was another reason why this lipid was not formed in sufficient quantities. Now, I designed another system where some of these issues related to stability were addressed. So I came up with a lipid synthesis scheme, which is somewhat related. So here I took an enzyme known as FADD2. This is also from Mycobacterium tuberculosis. Now, instead of an fatty acyl adenylate ligase, this is a fatty acyl CoA ligase. And what this enzyme does is it takes a fatty acid and converts it to a fatty acyl CoA with ATP and CoA and magnesium. Now, the fatty acyl CoA is hydrolytically more stable than a fatty acyl adenylate. And also, it can react very selectively with a lipid fragment which has a cysteine group on it. Now, this reaction is also taken from peptide ligation chemistries. There it's known as native chemical ligation. Now, these two can react very selectively and rapidly to form these amide containing lipids. So, in this reaction scheme, I mentioned that the fatty acid CoA had improved hydrolytic stability, and that's why it reached a higher steady state concentration. And also, this linkage, this is an amide linkage, this is also hydrolytically stable. So the issue of hydrolysis that we saw with the lysolipid in the previous example was also solved here. So this two-chain lipid was generated in very high yield when we added the DNA that codes for FADD2 to pure system. So FADD2 got expressed and it drove lipid synthesis in the manner that I described. And this in situ formed two-chain lipid self-assembled to form vesicles in the pure system as well. So then I wanted to introduce more complexity in the picture. So what I did is that I tried to co-express another protein with it. Now, because this lipid that we were generating, it was a modified sphingomyelin, I chose to work with a protein known as lysine, which has some natural binding affinity towards sphingomyelin. So here I found that lysine, in spite of the modification, the cysteine modification on the sphingolipid, can bind to the head groups of this lipid. So this is what I did, and this is a little bit complicated experiment, but I will try to break it down in a simplified way. So here we took the pure system, we added the DNA that codes for FADD10, we added the DNA that codes for SFPFP anti-lysine. Anti stands for non-toxic, because lysine is otherwise a toxic protein. And then we also added the lipid synthesis precursor that I described in the previous slide. And then FADD2 got expressed, and then it drove the synthesis of the two-chain lipids. The lipid got self-assembled into vesicles, and then on these vesicle membranes, the SFGFP lysine that got co-expressed, they went and bound and decorated the surface of the vesicle. Now, all of this took in a one-pot fashion. So lipid synthesis, transcription, translation, lipid synthesis, and self-assembly of those lipids into a vesicle, and having those vesicles decorated by a specific protein. So with this, I would like to summarize my second story. So this story started with a fundamental question, and that question was how first complex membranes were synthesized. And this problem was attempted by carrying out reactions between two lipid fragments. So this was done to mimic the action of acyl transferase enzymes. And then we brought in a soluble enzyme in this picture and had those single chain amplifiers react between each other to form these two chain lipids. And then we brought more complexity in this picture. We had that enzyme expressed in a cell-free protein expression system and drive lipid synthesis in presence of the lipid precursors and synthesize vesicles which were decorated with a specific kind of protein. Now I will briefly touch
I think we lost our speaker. I will text him and see if he can reconnect. All right, we're back.
Sure. Okay. Yeah. So yeah. So
yeah, we heard this slide, and I think you dropped off right when you switched to the next
with this
slide.
Theme of chemical synthetic biology. Okay. Okay. So I continued with this theme of artificial cells and chemical synthetic biology later in my so here, just as a smooth transition from artificial cells, I moved into the world of viruses. So I thought that viruses could also be a way to address the fundamental questions like what is living and what is non-living, and what distinguishes living from non-living. So this took me to the lab of Professor Stephen Boxer at Stanford University, where I studied the fusion mechanisms of viruses. Now, when I chose to work with viruses, this was still before the COVID-19 pandemic. And coincidentally, once I started working with viruses, I also realized that no, I should work on some problems which are related to human health. That was not my initial motivation. But then I developed some assays with influenza virus, where I developed a method which could be used for to probe, transfer, and distribution of viral content during fusion. And this mimics an actual cellular infection in some fundamental ways. That's why this way of doing fusion between a virus and a vesicle is referred to as a pseudo infection. So in this method, I was able to show how much of viral contents get transferred to a target vesicle. So in a way, this is mimicking endosomal escape. And then a next step will be to see whether this kind of a methodology can be applied to other viruses and also other kinds of nanocarriers which are inspired by viruses. And in the beginning, I made a passing reference to the archaeolipids that I worked with. So these are very interesting looking lipids that are found in nature. So they have these highly branched hydrocarbon chains. And in contrast to bacterial or eukaryotic lipids, which have um, ester linkages at their head groups, they have ether linkages, which makes them chemically very stable. And they also typically lack unsaturation. So because of all these reasons, these lipids allow archaea to thrive in very extreme conditions like Yellowstone National Park. So if you have been there, you have seen this boiling lakes and like lakes which have very acidic water, like the pH is probably 1 there, and all those places are thriving with microbial life. So these kind of lipids offer adaptation to them, to such harsh conditions. So in this work, I studied the structure and function relationships in archaeal bipolar lipids and offered some idea about how this kind of organisms may survive in such environments. And finally, I will wrap up my talk by giving you some kind of insight or perspective on what we learned from this artificial cell or chemical synthetic biology approach. So first of all, the chemical synthetic biology approach can be used to address many fundamental questions on origins and evolution of life. For example, how the earliest complex membrane lipids were synthesized. Then this whole idea of making an artificial cell is a very creative pursuit. So it often leads to many unexpected discoveries. So I briefly mentioned one reaction that I discovered when I was doing some of these reactions between lipids. So this is a copper catalyzed O acylation of 1,2-amino alcohols. And through this reaction, I discovered a unique isomer of sphingomyelin, which was not reported before. Then, while we try to make artificial cells, we discovered new interesting materials, such as the lipidic sponge phase, and also many new vesicle-forming lipids. We can also discover new phenomena, such as the example I gave on shape transformation of droplets. And similarly, there are many other discoveries that can be made along the way as we try to make these life mimetic systems or so-called artificial cells. So with this, I would like to thank all the individuals who were integral to my scientific journey. Like most importantly, my advisors, my PhD advisor, Professor Neil Devraj, and my postdoctoral advisor, Professor Stephen Boxer. I also want to thank Professor Sunil Sinha, with whom I collaborated during my PhD. And because of him, I got interested in X-ray scattering and biophysical chemistry in general. Then I want to thank Henrike Niedel-Holtmeier, who was a very good collaborator of mine during my PhD, and she's a professor at TU Munich right now. And then I want to thank all other collaborators and my former lab members who really encouraged me, who were part and parcel of my scientific journey. And finally, a very special thanks to Kate again for inviting me to speak at this very nice community. So thank you very much. Thank you. My thanks to the audience for your kind attention, and I'll be happy to take some questions. And I will end with some science artwork. So because I work with vesicles, I often see very interesting patterns under the microscope. And I try to imagine some shapes, some kind of faces, or some kind of objects within them. So I did a little bit of coloring using this image J. And I also gave some names to this. So these are some of the things which are actually lipid vesicles that I observed under the microscope. So yeah, thank you again, and I'll be happy to take some questions.
That is awesome. I would wear a t-shirt with some of those printed on it.
Yeah.
There is a lot of questions in chat. Can you see chat?
Okay, let's see.
If you can read a question out loud before answering, that's going to go for the recording.
Sure. Yeah, sure. Okay. So Hannah said, thank you for a great talk. What's the diversity of lipids you can make in situ? For example, different head group modifications. Okay. So I will answer this question. Yes, it is different head groups, lipids with different head groups have indeed been made. Not that much in my research. I think we were mostly working with phosphocholine containing head groups. In one example, I did make a phosphoglycerol containing lipid, but there have been very involved studies where bacterial lipid synthesis pathways, which can make different head groups, were reconstituted inside of vesicles. And those kind of like quite a bit of diversity in terms of head groups were shown. Okay, so the next question is by Ben Arias Almeida. So Ben is asking, I want to ask you about your work on archaeal lipid synthesis. Can you elaborate further on the process used for their synthesis? Do you know if their conformation was through the membrane or U-shaped? Okay, yeah, that's a great question. Thank you, Ben. So these lipids that I showed here, these were actually made through total synthesis. And I didn't do the synthesis myself because it's a very difficult job. So this was done by our collaborator, um, Isaac Falk from Noah Burns's group. So they're a specialist in organic synthesis. So made this lipids through, I believe, at least 15, 16 steps. And then coming to this question of whether these lipids form U-shape or they're completely straight in terms of conformation. So in this paper, we actually addressed this question. So what we did is that it's not so easy to just look at the membrane and find out whether these lipids can fold into U-shape or they're completely stretched because they're too small. How do we know that? So we looked at some kind of functional behavior of the membrane that may arise if these lipids have U-shaped versus stretched conformation. So if these lipids have U-shaped, or at least some of these lipids have U-shaped, then there will be regions in the membranes which are bilayer in structure. Now, a bilayer structure is essential for membrane fusion to take place. Like if you look at the classical model of membrane fusion, then you will see that fusion takes place through bilayer regions of the membrane. So in the first step, the outer leaflet merges with the outer leaflet of another membrane, and then a fusion pore opens up, and then the inner leaflets merge. So that's how fusion takes place. So we showed that these vesicles can actually undergo fusion. So that is one evidence, a functional evidence that these membranes must have some regions which have U-shaped lipids. So maybe some of the lipids are also straight and they form something known as a monolayer, but there must be some regions in the membranes which have these U-shaped and bilayer regions through which important processes like fusion can take place.
I realize that you probably don't see a lot of questions because when you dropped off the call,
Oh, they
you
just
lost the chat.
disappeared.
Yeah.
Oh, I
So
see.
let me read.
Hmm.
Let me start reading them from the beginning. Um,
Sure.
so Rooney asked, what is
Hmm.
the salt tolerance of the droplets? Can they tolerate higher mono and divalent cation concentrations?
Yes, they can. So these droplets are made from non-ionic amplifiers. So they really don't interact very strongly with divalent cations. So in this paper, we showed up to 10 millimolar, I think, I believe it was 10 millimolar calcium that it could tolerate. With magnesium, there wasn't much of an issue. And with the sodium chloride, these droplets are stable over a very high concentration of sodium chloride. I can remember we definitely tried 1 molar and the droplets were fine.
Right.
Hmm.
Stacy is asking, could you comment about possible interactions between your lipid droplets and peptide coacervates from intrinsically disordered proteins? The
Oh, that's
coacervates
a
are explored as a simple compartmentalization tool. I wonder if there is a way to combine those two systems.
oh, that's a beautiful question. No, we haven't tried that, but it will be very interesting to explore, like whether they form some kind of a hybrid material, whether they just like each other or not, or whether they have some kind of uh, positive or negative interactions between them. We don't know, but that's certainly a very interesting thing to explore. I know
Fenguan is
that there
asking,
have been
oh,
studies where people tried assembling, I think Christine Keating did that.
oh
They
yes.
tried assembling vesicles on top of this peptide or
Yeah.
protein kind of coacervate. Yeah.
I just a few months ago, I had a paper with Chris Keating on doing exactly that. We
Oh, I
synthesized,
see.
yeah,
Yeah,
we
I think I
synthesized
saw
the
that. Yeah, yeah, I think
coacervates.
I saw. Yeah, very
Yes,
nice.
I would be really curious to follow up on that.
Yeah, yeah.
Okay, next question. Fenguan is
Mhm.
asking, great talk. For the lipid synthesis, is
Thank
it
you.
possible to increase the yields using whole cell extract, not pure? I would imagine the
Hmm.
yields would improve significantly.
Yeah, that's a great idea. The reason why we work with pure system is that the pure system has much fewer things. So it's much easier for the in situ form vesicles to assemble, in situ form lipids to assemble into vesicles. So I think one difficulty in carrying out the lipid synthesis in whole cell lysate will be that because it's so heterogeneous and it's quite viscous, it's so rich in proteins that the in situ formed lipids may not assemble very well into vesicles, even though the lipid synthesis may be better. I mean, I'm just guessing, but those lipids may just get aggregated. And also the other reason why we use pure system is that it's very well defined. So it's very easy to uh, control the composition of the system, because when we did the lipid synthesis, we had to add a lot of extra things. We had to add fatty acid, isolipid, and then some ATP, CoA, etc. So anything you add to pure system actually perturbs the pure system, and it affects the performance of it. So at least if we know exactly what's in there, it's easier to optimize uh, the system. So that was the reason. But I think like as you suggested, the lipid synthesis part may be better in wholesale lysate, but I can imagine that it will be more difficult to form the vesicle.
Yep.
Hmm.
Nina says, awesome project. Thank you for presenting. Do those newly
Thank you.
synthesized lipids flip-flop to the outer layer, or do they stay in the inner leaflet?
Okay. So this is a bit difficult to answer, but we, like workers from the Raj lab, we do have some evidence to suggest that when these lipids are formed in situ on the membranes, they may flip-flop to the other side, because these transient intermediates that are formed, it's a little bit difficult to characterize and know what's exactly going on. But based from the functional um, behavior of these membranes, we have good reasons to think that the lipids may be able to flip-flop while they're getting synthesized.
Yep. And oh, I think you read Hannah's already. Okay, so
Yes.
we're down to the end of the questions. Yeah, I would like to follow up. I really like the question about the meshing that system with the coacervat systems, because that's something we're working on in TXTL too, and I would love to see some sort of a hybrid.
I see.
I think that would be really powerful. All right, last call for questions. Going once. And I want to say thank you again for a really inspiring talk. I really like to see when those seminars
Okay, thank you very much, Kate.
Talk about origins as much as synthetic biology.
Yeah. Yeah, I also want to thank you for inviting me, and it's very inspiring that you are continuing this community for quite a long time. And I do have some like wish for starting a community like this in India. The synthetic community is still not that big in India. I think maybe 20 to 30 labs are working on this kind of thing. But it would be nice to have a community here as well. And surely, I hope I can get some inspiration from you. If I want some ideas or advice from you, I will reach out to you.
It would be fantastic if you could
get
that started. There is a really strong Asian synbio community going, so it would be great.
Okay.
All right. Thank you so much, and thanks everyone.
Okay. Yeah.
And have a great rest of your Monday. Or good night to you.
Yes.
Bye.